Hey, hello everyone. Good evening. Welcome. Hope all of you are able to see my screen and hope all of you can hear me. Can I have a quick confirmation, guys? Perfect. Thanks for the confirmation. Guys, please remember this. I have already informed this in the previous week. So we have classes on alternate days. That is, we have classes on Monday, Wednesday, and Friday, right? And every day it is going to be 90 minutes. So since we are in the beginning of the classes, I'm not taking continuous 90 minutes. Instead, I'm taking it for 60 minutes duration only. Okay, because these are going to be like most of the theory that we are discussing. It would be difficult to continue for 90 minutes of the time. That's the reason approximately between 60 and 75 minutes, I'm stopping the session, right? And if required, sometimes let's say that we need some continuity. In that particular time, I'll inform you one day prior. Let's say that uh, today is Wednesday and tomorrow also I would like to continue because I want to finish few of the stuff which is really important. In that particular case, today itself I'll inform that hey, tomorrow we have a class and we can continue, right? So this is how we are going to proceed. Make sure it is going to be Monday, Wednesday, Friday regular classes. Sometimes we may take additional classes on Tuesday and Thursday. If that is the case, I'll inform in prior. Am I clear with that? Cool. Now, so last week we have completed all the basic information that we required to start with our Sdtm, right? So we already informed you. First, initial things are going to be we understand what is called clinical trials. That's the first thing. We understand all the phases of clinical trial, and then so in the clinical trial, the most important thing is going to be how to kickstart this and what are the various definitions. As part of this, initially we started with a protocol, the protocol document, and we have elements of that particular protocol. We have discussed them as well. In addition to that, we have also discussed about few of the definitions. Correct? So these are the things we have discussed, and then on I think Monday, I have informed that we are going to kickstart SDTM in the next class, that is today's class. So first we continue with SDTM, and then we have ADaM, and finally we go with the tables, listings, and figures, which is nothing but the TLF. This is going to be the flow. All right. So let's not waste any time. So I don't repeat this one now because all of you are pretty clear with this particular one, clear your clinical trials. I also believe that all of you are pretty comfortable with protocol and its elements. The definitions was the one which we discussed in the previous, this one. In the previous one, let's discuss that once, not discuss, just quickly we go with this particular one. So initial thing we discussed was blinding. So anyone tell me what is called blinding now? Blinding or masking is nothing but what information we are not disclosing, what information we are actually not disclosing to the people. So if we are disclosing everything to everyone, because we have three different entities involved, what are they? Subject, which is nothing but the patient. Then we have investigator. Finally, we have the team who is actually preparing all your data, analyzing the data. If we are disclosing all the information to all the three people, we simply call it as a unblinded. Correct? Yes, exactly. If if I'm not disclosing information to the patient, but investigator and team is aware of that particular one, in that particular case, I'll call that as a single blinded. Correct? And what is called double blinded? Anyone? Someone responded no. Okay, it's okay. So and single blinded is the one where either the patient sorry, single blinded we already done. Double blinded. A double blinded is the one where both the patient, right, and also the investigator not aware of anything. right? Sometimes investigator might aware of this, but we can also have a case where patient and the team is not aware of what is the drug that they are taking or the drug disclosure. Correct? So both the cases will fall under the double blinded. For sure, patient does not aware of what the treatment that he is undergoing, but either of the investigator or the what to say, the team is not aware of the drug information or treatment information. That will call as a, just give me a second, this is something important. Okay, sorry guys, some important call. That's the reason I supposed to attend it, and it's done. Okay, I switched off mobile, so no issues here after. All right. So double blinded is also done, and the third and the last one is going to be the triple blinded. The triple blinded is the one where none of the people who are part of this particular approach are not aware of what treatment or what drug which is actually happening there. So they're strictly going to follow the protocol. As per the protocol, they're going to proceed. Correct? So this is what the first thing, that is blinding. So I have given all these details here. You can refer later. The second thing is going to be ICF. So ICF is nothing but anyone, what is called ICF? Can someone please mention about what is called ICF, the acronym? Wonderful, Ankita. Informed consent form. This is the form which is going to be a, I can say that a legal document which we actually get it from the volunteer or the subject, which is having it, which is a signed form, and the most important thing is going to be we are going to capture the date there. Correct? So this is a legal form which has all the inclusions, exclusions, all the details will be available there. Correct? So why we need this particular one? The informed consent form, to make sure the subject is well aware of what is happening there and to maintain the integrity. Now, what is the next thing? Screening. Screening is nothing but, so let's say we are volunteering, and before we go with this before we pick this particular person, right, we need to ensure that the subject is actually fit for this particular clinical trials or not. So we have set of inclusion criteria as well as the exclusion criteria. So we need to check all these inclusion or exclusion criteria are meeting or not, whether we can consider him or her as a subject or not. So that can be called it as a screening. The next thing is going to be randomization. What do you mean by randomization? Randomization is the process where, so we have a lot of volunteers, or we are going to have a lot of subjects, so many subjects. So we are going to have the right mix of both the people. We need to say, let's say I have 100 volunteers. Out of them, we are going to pick 50 healthy people, or 60 healthy people, or 40 people who are actually the patients who are suffering with the other diseases, or the same diseases. So how to randomly pick the people? So we have a population, we have a sample. How to get the right sample is nothing but yeah, randomization. Randomization plays a very important role while selecting the subjects. And then, so we also talked about two things, that is nothing but control group, because we are going to do the grouping. So control group is the one, and another thing is going to be, what is the other one, guys? Anyone?
Treatment group.
What is the other name for it? Anyone? Exactly. Either you can call it as a it is intervention, not the investigation, but few books also refer that particular one, because they are using the investigation drug. We are actually providing, we are actually giving the investigation drug to that particular people, or the study drug to that particular people. So the people who are actually taking the drug that we are testing is nothing but investigation intervention group, or we can also call it as a study group, whereas the control group is the one where the people is not taking the study drug at all. Actually, they are taking the placebo. Placebo is nothing but a fake pill. Correct? It doesn't have any active elements. It is just a fake pill. Correct? So we also talked about treatment, follow-up, and proceed. What is the procedure? The procedure is going to be, first we get the consent, then we do the screening, randomly we create the groups, then we provide the treatment, and we do the follow-up. Correct? And then In each clinical trial, there are phases. Each phase is nothing but an element. Group of elements together can be called as the approach. Scheduled visits and unscheduled visits. What is scheduled visit? As per the plan, as per the prescription, we are taking it. Correct? Intervention, act 2 or treatment group. Perfect. All the three are, we can call it. Even investigation also. Investigation group also is the right name. Thank you. Thanks for sharing the details. So these are the various definitions that we would like to understand before we start with the SDTM. So hope all of you are clear with this. Now, guys, did you ever get a chance to read the protocol now? Because you know all the elements, you know all the things now. So if you go now and if you read any of your protocol, let's say this is one of the protocol. This is Alzheimer's disease protocol, correct? Not completely, at least most of the information you might aware of that now. So phase 3, so that the current element is going to be phase 3 element. Double blinded. What do you mean by double blinded now? The double blinded is nothing but patient is not aware of it and investigator or the analyzing team is not aware of what is the treatment that is happening. Placebo control. What do you mean by placebo control? So there is also a fake bill which is supplying along with this particular NS0314D drug. Both of them we are giving them. Correct? Right. So what is the name of the investigational drug now? NS0314D. So since already we mentioned the phase 3, here you can see the phase 3. So now you can see objective, the primary objective, secondary objective, what is called a safety, endpoints, lot of design. You can see the trial design. Under the trial design, you know it is going to be a parallel group. It is going to be a parallel group. And it is randomized. Randomly, we are picking the people. Multi-center within the same nation, but it is going to be at the it is multi-center as well as the multinational, meaning across the globe the trial is happening as per the protocol. Within each country at multiple centers it is happening. Clear? So what exactly it is happening now here? Now you can see almost what is going to be the trial. All of you are able to understand now. So before we may or may not aware of all these terms, but now when you look at this particular trial design, when you look at this particular document, we can understand what exactly is happening. Am I correct? Can I have a quick confirmation? Wonderful. So all of you are pretty clear with this particular one. So I'll share this particular one, guys. Just have a look into this particular one so that you can understand what is going to happen there. Now you can see the screening, double-blinded treatment, when the visits are there, randomization when it is happening, when is the end of the treatment, all these details you can see. Correct? And I also told you the inclusion as well as the exclusion criteria. You can read them. Right? It is not same. Why we haven't discussed inclusion and exclusion criteria in detail means because it is different from each drug to other. At the same time, this criteria is different from one treatment to the other treatment. Correct? So that is the reason we are not discussing about the inclusion as well as the exclusion criteria in detail because it is completely random and it is vary from. Okay. All right. Now. This is the, I think I have already shared this particular one with you. If not, I'll share it today with Melissa so you can read this particular one. Now, come back to the next one. So protocol definitions we are clear. So today we are going to start with the SDTM introduction. So before we go with the SDTM introduction, may I know like who actually can I say who might have actually created this particular one, SDTM? Anyone? Okay, why SDTM has come to the picture? Why do you need it? Initial classes for standardization. Wonderful. Okay. Okay. Let me just reiterate that particular fact and we can proceed there. So what we understand, every drug that we are actually producing, that can be a treatment drug, that can be a vaccination, or that can be a medical device or equipment, need to be tested for two things. What are them? Safety and efficacy. Correct? So to do that particular, what this governing or the authorities are going to do, the authorities are the one who need to approve them. They need to give a go ahead, then only it will come to the market. So let's say one of the regulatory authority is FDA, correct? So now, which is as part of your United States of America. Now, so we know that each company can produce their report to get the approval to the FDA. If the reports are not standardized, if the reports are not following the same format or same structure, it is difficult for FDA to approve them, to at least to analyze them, correct? To review that, if all of them are following the same format, it is easy, quick to review, and we can also do the automation there. I mean to say that we can also implement the machine learning models there, right? So FDA is looking for the standardization. So what FDA did is, since they want to look for that, since they are looking for the standardization, so they have come up with an organization. They have created one organization, right? And that organization, any idea what is that organization is? Anyone? That organization is nothing but the CDISC, right? So we all know how our CDISC is nothing but global. It is universal. It is everyone can use this particular one. I can simply say that CDISC is one of the organization. Here you can see, correct? So we have all the details. You have standards. If you want to go for the, even CDISC is going to provide all the education as well, but it is a bit expensive, and I can say that at a very high level it is going to be. It is not going to deal in detail, right? So this is what your CDISC is going to be. This organization, you can also call it as a, this organization main purpose is to set and maintain the standards, correct? I need to set and maintain the standards. At the same time, the standards need to be easy to understand, and also we need to provide the guidance for these particular standards. That is the whole objective of bringing your CDISC, correct? So CDISC, where it is, just a second, here it is. Here is what your CDISC is. Now the CDISC has come up with three different approaches. There are a lot of approaches, but we are interested in three. Out of that, the very first one is going to be Sdtm, right? There are many approaches, but what we are very much interested in is going to be a Sdtm, correct? So I hope all of you know the acronyms for Sdtm as well. Anyone? Study design, or I can say that standard design, tabulation. Anyone want to finish this? Exactly. Study design tabulation model, or you can also call it as a standard design tabulation model. And we also have ADaM that we can discuss later, and we also have your ALF. So today we are going to start our discussion with Sdtm. So today we understand what is going to be the end-to-end workflow. So remember guys, all of you will not get a chance to work on all these three steps or all these three different modules, right? Some of you, once you join an organization, you may stick only to the Sdtm. Some of you might be working only on ADaM. Some of you might be working on your TLF, right? Some of you might get chance on working on more than one model as well, or more than one design as well, right? Anyways, we are going to understand the complete flow, and we are going to work on the complete flow as part of the project. But today we will be specifically starting with the Sdtm. All right. So here we also need to understand few things. I think whatever I have done so far, I have explained this, because all the clinical trials are usually straightforward. All we are going to check is safety and efficacy. All these things we are going to record. All these things we are going to collect. Indirectly, I can say that there is going to be data driven. So the entire thing, the entire approvals, everything is completely based on data. The data need to be recorded, the data need to be collected, it need to be compiled and analyzed. So how properly you're handling this particular data will ensure how nicely or how perfectly you are actually producing your reports. So to maintain the data properly, FDA has come up with the CDISC, correct? So as part of the CDISC, you can see the CDISC standards here. So there are 3 people involved. Who are the people involved there? Researchers, pharma companies, technology vendors like us. Okay, how these CDISC standards are helpful to us? I can say that, let's say if you're a researcher, simply I can say that how the data is. We are going to have a clear, that is the cleansed data and simple data management at all stages of your clinical research, correct? So what is going to happen? The results will be quick, the outcomes can be easy, and it will be, I can say that, simple to derive, simple to understand, simple to produce. Because researcher is the one who is going to give you the breakthrough in your clinical trials. So if the data is clear and simple, if I have effective data management, my outcomes are going to be quick. So from researcher's point of view, it is more about my data. From pharma companies never bother about how the research is happening, all those. Mean to say that in detail. All they need is, I have a report, how quick my report is going to be get approved. So that if the report is approved, I can send this particular drug to the market and I'll get benefit out of it. Correct? So all they need is how simple and fast I can submit my report, and how quick my CDISC is going to approve it, and how quick I'm going to deliver this particular thing to the market. So technology vendors like us, right? So I already have the data sets. I already have all the data sets. How quickly I can create the solutions, how quickly I can produce the analysis on top of it, right? At the end of the day, all we need is, we have millions and billions of the data. All these millions and billions of the data that we have extracted based on the subject's information, that need to be properly processed, that need to be collected, that need to be analyzed. That's the whole point of bringing the CDISC into the picture, correct? Now, in the CDISC, before I go with CRF and all, let's draw a simple workflow. What is going to be happening there, right? So the first and foremost thing. So first we are going to start with a very basic thing. We first start with a clinical study. Let's say that I can call it as a study itself, correct? I have my study. I want to start this particular study. Okay, so we have started this. What's next? Once I have started this particular study, that is nothing but, study is the one where I have all the phase 1 preclinical trials, phase 1, phase 2, phase 3, phase 4, all the things are here, all of them. So to do this particular one, or once I have this particular one, to proceed with the next step in this one will be, I need review documents. Most important thing, guys, try to understand, ask me as many questions as possible. Okay, all I'm looking is, my second step after my study, or after I kick off my study, is going to be, I need to have the documents. If someone asks me, Murli, what are the documents that you are looking at? I can say that the documents that I'm looking at here is nothing but, we generally call it as review documents. So what are those review documents are? The review documents are, number one, I need protocol. We discussed this last week, right? Protocol is going to be the first document which is going to kick off your clinical trials, because it has everything, right? Okay, I have protocol. What's next? I need to have CRF. CRF stands for case report form. Let's discuss about what is this case report form is and how it looks like. So this is your case report form template. This is a blank template, guys, right? You can fill it up. So a case report form is the most important thing, because it has all the information of an individual study, right? Based on the protocol, what and all we are doing, all those information will be available here. Why it is important? First thing, to ensure the data collection in accordance with the study protocol. So what do you mean by study protocol? Your protocol will say that, hey, you need to collect this particular data like this. So I'm going with the parallel study design. And in the parallel study design, this is the dosage that I would like to provide. This is going to be the period that I want to provide. So these many random samples I'm taking. And also this is going to be, let's say, the double-blinded one. According to that, I need to get the data. So we need to collect the data as per this particular one. Again, what and all the vitals that I want to collect. Those information also I need to do. Second, whatever I'm filling there, right? So I need to ensure that the data collection should be in compliance with the regulatory authorities. That can be FDA, EMA, or our drug control, whatever it is. It needs to be as per the regulatory authorities, right? And most important thing to be, I need to make sure that it should be effective, the data collection should be effective, and I need to ensure that the reporting is proper, and main thing is going to be, I need to promote the safety of data sharing between the various teams and departments. All these things need to be included in this particular CRF form. In general, what exactly this CRF form is actually including? Study title and the number, who is going to be the investigator, what is going to be the patient or subject ID, inclusion or exclusion criteria, demographic data, detailed description of dosage, how much dosage and what is going to be the and how frequently I need to give that particular one. And also you can see the remaining things, the investigator signature, date, and everything. You can simply see this one, it is easy to understand. I can have the study title, you can see this, and then we are going to have the where the site is nothing but the hospital or clinic or doctor's home, whatever it is, who is going to be the principal investigator, the subject initials, subject randomization number, that's nothing here, ID, investigator signature and date signature, you can see here. Inclusion criteria, what and all I'm including, see this. If the subject is healthy male aged between 18 to 60 years, yes or no, right? So exclusion criteria, and you can see that the ICF, the consent, the demographic data is nothing but age, sex, and your height, weight, BMI, all those. You can see here all the demographic information, right? So screening, I told you the screening is very important before we get this particular person is a fit for my clinical trials or not. See the previous medical history. This is going to be around 15 pages of the document, and this 15 pages of the document will have all the information that a subject is actually screening and undergoing, right? So this is, now you can see that, now you can understand why it is important. This is important because it has each and every information about the subject, individual subject. Remember, this is not at the whole clinical trial. It is not about the drug, it is about the subject. This is about the patient. You understand, guys? How do I differentiate it? Clinical trials is a whole. So to start this particular clinical trial, I need a subject. The subject is nothing but the patient. I'm collecting all the patient information. Yeah. Is that clear? Can I have a quick confirmation? So once I have this particular information, now after CRF, I need something else that is nothing but a little advanced of this. We can call it as ACRF, annotated case reported form. That is, we just have annotations there. So what is that? I'm going to show you anyways. In addition to that, we also need SOP, standard operating procedures, right? In addition to that, we also need SAP. This is nothing but statistical analysis procedures. That's nothing but statistically what and all we want to conclude all those information. But we mostly required these four. What are them? Once before we want to start with the next process, I need to have the details about what is the protocol. That is the first thing. It has information about the complete clinical trials that we want to execute. CRF, a case report form, which is very specific to the individual subject. Annotated CRF, I'm going to explain what exactly it is. And then standard operating procedures, it varies from subject to subject and clinical trial to clinical trial. Statistical analysis procedures, these are also going to be varies from subject to the subject. But on a whole, I need all these documents. Let's discuss about how this ACRF looks like. ACRF, which plays a most important role because it is a blank CRF. First thing is it is a blank, right? And it also have annotations about the document, the location of the data with respect to the data sets. I forgot to add one more. I talked about all this, but I haven't talked about the most important thing, which is nothing but raw data sets. All your clinical data sets need to be provided to you, right? It should be looks like this. Let's say someone has given you the data like this. These are going to be all your SAS data sets. Can you see this? AE, CM, TA, DM. I have renamed each of the SAS data sets based on the domain. Guys, these are all the domains. Demographic data, you have AE, CM, CO, DA, PR, PQS, all these are going to be the domains. We'll discuss all the domains, don't worry. Right? Now, each of these raw data sets are going to be the input to me. Along with that, I also have my CR. I also have my CRF, case report form. I also have metadata. That is nothing but three things I'm taking here. What are they? Number one, or simply we can take two. I have CRF, which has information about the individual subject. Number two, I have my what is the other one? Raw data set. Let's say a simple way. Here I have subject demographic information. Here, let's say age, sex is there. I want to know where this particular field is, right? Because I have a lot of data sets here, right? I have data set called AE, CM, DD, DM. Where exactly this particular field I can see, or where exactly I can identify, I can find this particular field. Even if I identify that particular field, with what name it is going to be, because I have here body mass index. SAS variables does not have any spaces. We are going to maintain our SAS variable names in compliance with the SAS variable regulations. Correct? Your variable name should not have more than 32 characters long. Your variable should not have any other special characters other than the underscore. Your variable name should not start with a number. It can be either alphabet or underscore. Correct? We have a lot of rules for defining your variable name. Correct? So now the biggest task is going to be, if this is going to be the label, I can call it as a label or a parameter, where this particular parameter is exists, in which data set it is exists. If it is exists in this particular database or in this particular table, with what name it exists, I need that information. That information will be part of your ACRF. This is called your annotated CRF. Can you see this? Right? Now, can you see this particular one here? Let's say, so here I have few things. What is the first thing? The first thing is going to be prior or post-study therapy. One more is their standard implementation rate. Sure. That is also required. I'll go one after the other because that is the guide which is provided by the CDISC. Correct? So I'll also give that information. Currently 3.3 is going on, but most of us are still following SDTM implementation guide 3.2 version. I'll also give that. So now you can see that if I want to know the information about prior or post-study therapy, if I need this particular information, this information is part of AC table, that is AC data set. Here you can see the data set, right? One of the data set is AC. I don't have all here, but don't worry. In the AC data set, there is a name, there is a variable name called ACPRIPOS. This is going to be the variable name. This is going to be the table name, and that has information about prior or post study therapy. So if you go with CRF have only the name. But when it comes to the ACRF, it also have the details about which location, that is nothing but where exactly you have this particular one, right? But one problem with ACRF is ACRF has every part of every piece of information that may be submitted, may not be submitted. In this particular case, it is not required, so we have not submitted this. We have not submitted this, right? Now you can see the therapy type, what therapy type it is. The therapy type can be, again, it is part of AC. You can call it as ACTYP. So this is going to be CEM's SCAT. That is the value of it. Correct? Can you see this? All the information, whatever the information is going to be. So here I have adverse events, AE. So we have the AE data set here. You can see the very first data set itself is the adverse event data set. So this adverse event data set has the information about this. Can you see? What is the adverse event? What is the name of it? Now you can see adverse event is part of the AE dataset. The name is going to be AE term. Is it ongoing or not? What is going to be the start date? What is going to be the end date? Right? What is the severity? You can see the information. Right? Let's see one more table. Let's see demographic table. Where it is. It is also AE. What's SDV again? This is the label. I'll give this information later. This is nothing but the label, right? So you're going to have this throughout, not here, throughout you're going to have it, right? So this is nothing but this particular study details name. So that's the reason it is going to have throughout, completely you're going to have the same name, right? So this is varying from one to other, so we don't need to worry much about it. All we need to worry about it, we need three things. The first one, what is the parameter you are looking at? Number two, what is the column name, where it exists? These two are the most important thing. There is another thing we can call it as in your protocol, in which page it is, or in protocol or in your CRF where it is. When it comes to the in protocol where it is, in CRF where it is. That is the orientation. I can show you that. I can show you that. So first of all, can you see what is called CRF, ACRF, and how it is different from CRF? Can I have a quick confirmation? So I have two things now: CRF, case report form, annotated case report form. How these two are different? All of you are able to understand? This is very important, and also this whole ACRF will be reviewed by the team leader. So in every organization, we are going to have a team lead. The main use or the main thing that is going to happen here is, maybe I need to tweak this a little. I'll remove this. Based on this particular information, we are going to create a ACRF. This ACRF, whatever we are talking, okay, there is a question to me. Are we covering this? How to create this ACRF? I can say that it is purely into the clinical data management team. They are the one who do this, because whatever we are doing here, I'll tell you where the SAS programmer will role will come. So generally this ACRF and all, we will not create. Mean to say that we, in the sense, SAS programmer will not create this. That's the reason I told you here, this ACRF is there, and this particular ACRF will also be validated by the team lead. But I'll tell you the process, maybe that can be helpful. Sanji, you are the one who asked, right? How it will be examined. I'll give you the process, right? Generally we don't do, but still I can help you with the process. So let me help you with the process. All right. So the process is going to be, I have included that here. You can see the approach. Always make sure that this particular annotated CRF, it is always to be in a Acrobat PDF format. Only Adobe Acrobat format only will be accepted because that is standard as well. And always it need to be included. The name should be always like blank crf.pdf. This is the standard. There are 3 steps in this particular one. There are 3 steps. Number 1, I need to determine which annotations are to be available in which pages and store them into an Excel file. Meaning, I'm going to refer this. This is my CRF, correct? I need to understand this particular CRF first. From this particular CRF, I'm going to create 3 different columns and I'm going to put it in an Excel sheet. Let me show you that. Here it is. Maybe I can zoom it a little. Yeah. Can you see this? The first one is going to be the CRF page. That is in CRF, where in which page this information is available. Next, what information you actually want to annotate, right? What information you actually want to annotate. Number 3, the orientation. Orientation is nothing but ranking. It has 4 values, 1, 2, 3, 4. That depending upon the what type of study that you are doing, based on that we are going to create this, right? So what is the first thing that we are going to do to go with this particular step? I need the CRF. This is the first thing. Number 2, I need to identify these variables. Variables are nothing but the columns, right? All these variable details are part of your SDTM. It should be somewhere. Just give me a second. This is one of these SDTM. Can I say that this is the version 3.2? In this, we have the details about the variables. I'll tell you where it is. Can I say this? Identifier, variables, these variable information I need to put it properly. So we have data sets and domains. These are the ones we are going to discuss. But can you see that the most important thing is going to be we need to know how to create the variables, what variables are there. Correct? So the first and foremost thing is going to be I need the information about your CRF. I need information about your variables, all the SDTM variables. Once these 2 are available, I can create them like this. This is the Excel that we need to create manually, right? So once you have this particular one, this Excel sheet, we are going to supply to the SAS tool. SDTM specification development means, where do you find it? Raja, you mean to say here, or I'm not sure what exactly it is. Okay, the specification development means where exactly the variable is going to be, how to bring it, all those information. Raja, if you want, you can unmute, you can talk about it. Else we can do this later also, not an issue. All right, let's discuss this and we can come back on it. So based on this particular one, because I want to create ACRF has 3 topics or 3 steps in that. The first step is going to be we are going to create an Excel. The Excel has 3 fields. In which page of my CRF I have the details. In which page? That is the first thing. Number 2, what demographic information I'm looking at. That I can achieve from my SDTM. Then the third thing is going to be the ranking, the orientation. This orientation is nothing but this, you can also call it as your priority, or you can also call it as a ranking. So there are 4 rankings has been given for each of this particular value based on each PDF that we are going with. The values are going to be 1, 2, 3, 4. There are 4 rankings, but how exactly the ranking is going to do and for what variables and for what domains, what ranking we are going to give, that we are not very keen into. We don't involve much there because just we are not the one who are going to produce it. Is that understandable? Why we are not going in deep means generally this we will not do. Being a SAS programmer, we will not do that. This can be done by your biostatistician along with your TL. We don't do that. Got it? That's the reason we are not interested to go into this one in detail, because the role of a SAS developer has not come yet. These are all not the roles of your SAS developer. We are not into that yet. Okay. So once we have this Excel we are going to supply to the SAS program. The SAS program is going to convert this Excel into XFDF file. The XFDF file is the one which is going to have all these boxes. Can you see this? These boxes, right? Annotation boxes are there, right? These boxes are for part of your file, and that file is called XFDF file. So then this XFDF file, there is going to be a process, nothing but what is going to be the color format, what is going to be the priority of this. The orientation will be decided by here. And also we are going to talk about which domain and all these stuff will be part of your XFDF file. There is going to be a separate program for this particular one, the SAS program. Few samples are with me. I'm going to provide those samples. But believe me, it is different from each CRF to other, and it may not be useful. But still, if you want to have a look at all these three, how I am giving this orientation details, right? And where exactly I need to place these particular annotation boxes, right? With what color I need to fill that. All those information we are actually programming by using the SAS itself. The SAS details, the SAS program details, I'm going to provide it to you. Just have a look. Is that clear? Everyone? This is not our piece, but still, if you are interested, I can provide the programming for you. Hold Sanchi and T, both of you, is that okay? Or do you have any follow-up questions on that? Anyone, any questions? Right. Remember, guys, the other part that I would like to talk here is there is a difference between the clinical SAS programmer and clinical research person. You can also call it as a clinical research associate. So this role, the clinical research associate, is the one, this particular guy, along with CDM team, clinical data management team, these two are responsible for producing this data, this ACF and all, ACRF and all. Being a SAS programmer, you're still not going to utilize this particular one. You still not go with this particular one. Or once you have ACRF ready, based on ACRF, your protocol, Sdtm, and raw data sets, then only you're going to proceed with your programming. I mean to say, maybe this is too early, but let's talk about it. There are 3 steps, guys. First thing is ACRF. Number 2, there is something called Sdtm mapping specification. Number 3, programming. This is called your SAS programming, purely SAS programming. So now we are here, that is nothing but we are at this particular box, ACRF. So let's say that your ACRF is good and it is properly validated. Okay, there is a question. ACRF annotation means how to write a specification file, how to write the programming. No. ACRF is going to be one of the input file to develop this Sdtm. Only when you have Sdtm mapping specification, then only you can start your SAS programming. Okay, let's start from here. Let's say I want to write a SAS program by utilizing your raw data sets, right? So let's say you're a programmer. So what are all the inputs you have? You have inputs about your SAS tool. Tool is there. We are all SAS programmers, we know how to use this. Number two, I have raw data sets. That is, whoever the one who is providing this particular project to you, the project manager or the team who has given all these SAS data sets to you. So these two are well aware. We know how the data set looks like, what contents we have there, and we have the SAS tool. But the thing is, how systematically I can use this particular data? Mean to say what variables I need to use, correct? And how to produce my TLF, the tables or listings or figures, which are part of your SDTM implementation, right? So currently 3.2 version is there. This is also with you. SDTM also with you. So SDTM has information about your variables. The next thing is going to be, let's say SDTM says that, hey, there is a variable called sex. Now, you need to understand where this particular variable will be. Because I have a lot of these data sets, right? In which variable it is, what format it is, right? What label we have, what is the length of this particular one, what is the source information. Only when you have this, it is easy for you to pick the right table and right information. Am I correct? Raja and everyone. Raja and everyone. Am I correct? You are a SAS programmer. I have given you the data sets. I have given you 100 data sets. Correct? But out of those 100 data set, from which data set you need to pick this particular variable? Correct? That information is part of this one. So this has the information about your SDTM, CRF plus your SDTM implementation guide plus CRF. These two information is part of this. Correct? Protocol, SDTM, CRF information. If I combine these three together, I'm going to get ACRF. But ACRF has information about table and a variable, correct? But it doesn't have information about the label. It doesn't have information about your format, the length, all those things, correct? To get those details, I need those details as well. Those details are part of your SDTM specifications. Is that? If I want all these information is part of SDTM specification Excel. And that Excel looks like this. Now this is metadata, just a second. Can you see this? Let's say I have demographic information. So the domain is DM. These are the variables. Study ID is the variable. The study ID variable information, I can get it from SDTM implementation guide. The label is called study identifier. You can see that it is a character. The length is 8. Code, is it required or not? It is required. And you can also see here CRF. It is actually the origin is a CRF, right? Domain is not part of your CRF. You are assigning this domain equal to DM, demographic domain. And you can also see that use subject ID, that is unique subject identifier. This is also unique. This is also going to be a derived variable. You are combining your study ID with subject number, right? Study identifier and your subject number, both of them you are actually combining, then only you are getting this. So are you deriving it? Are you getting this particular directly from the CRF? What exactly it is, right? So this mapping Excel, you can see that. What is the origin? Is it assigned one protocol or what exactly it is going to be? This information is actually part of your, what is that? ACRF. Correct? You can also see there is another domain. I have variable name, study ID, what type it is, what length it is, what identifier, is it origin or not, how it looks like. Can you see that all this information, right? This particular thing will be call it as your SDTM specification Excel. This SDTM specification Excel or this SDTM mapping specification Excel will be prepared based on your ACRF. So I am a SAS programmer. I have a tool, I have raw data set, I have implementation guide. But I need the mapping based on ACRF. So I need to provide it with these 5 things. SAS tool. Okay. How would we be preparing this SDTM specification by Excel? I mean, what will be the base to create this Excel? Apart from ACRF, will it be okay? Generally, you will not create this SDTM specification Excel from the base. You generally don't create, right? So who will we do this particular one? So to implement this particular SDTM specification Excel, you require ACRF, right? This will be provided by your TL, followed by you have your protocol based on the protocol document. You also going to have your SDTM implementation guide, and raw data sets. Based on this, we generally created. Let's say you are the one who started this particular project. Let's say that there is no prior history at all. There is no prior history at all, right? In that particular case, based on the CRF, because the CRF, what and all submitted, what and all not submitted, based on this. Let's say that. So I have this one action taking with this particular one, let's say AECAN1. Now you understand that AECAN1 is part of AE. So AECAN1, which is part of AE dataset, you are going to open the AE dataset and you are going to prepare what is the length of it, what is going to be the values of it. You are going to prepare this Excel on yourself based on you. You are the one who are going to produce it. In general, if it is prepared once, anyone can use it. So it is an incremental thing. It is not going to be created on one night or one day. No. Let's say there is a team. Let's say that you're working in a team. The team has a TL who is part of this particular project from past 2 years. So they already have created this particular one. You're simply gonna use this particular. Is that clear? You are not going to create this particular one from the scratch. No. If you want to do this, it is completely a manual process. This manual process can be done based on these 4 things. Am I clear? Now, this can be done by creating based on your blank CRF. First, you are going to use a blank CRF. You have your protocol. You have your Sdtm implementation guide, you have raw data sets. In addition to that, you also have CRF guidelines. So I told you right just now how to create this. There are 3 steps you are following. Based on the 3 steps, first you are going to create the ACRF. The ACRF will be validated by your TL. Once it is available, then you go to your SDTM mapping specification. To prepare SDTM mapping specification, you need these 4. Once you have it, then you will go ahead and you create your programming plan. Guys, is that clear so far? Can I have a quick confirmation? Right? Still, SAS programmer role is not here. So up to this, there is no SAS programmer. The SAS programmer role is about to start from SDTM mapping specification. If you are the one, there is no history available. Just a second. So first of all, ACRF, all of you are clear. We are going to have the blank CRF. The blank CRF based on the protocol, Sdtm guidelines, raw data sets, and CRF guidelines, we are going to create it. Once it is done, since either you are going to get the leverage on the previous guidelines that I have already created. If not, you are going to create from the base, that is based on the ACRF protocol, Sdtm raw datasets. You are going to create the guidelines like this. That is mapping, Sdtm mapping specifications. Once you have this particular specification, the next thing is going to be you are going to start a programming plan. It is not the programming yet, it is programming plan. So what do you mean by programming plan? A programming plan is that, let's say you are involved, let's say your protocol or your clinical research involved 5 different ones. TA, TE, TI, TS, TV. Let's say that. all the 5 domains cannot be given to you, and you cannot work on all the 5 domains at a time. So what needs to be done? Let's say we are 3 programmers. I can say that, hey, let's give TS and TV to Sanchi. TA I can work on myself, and let's say that give TA and TE information to, let's say, Manira. So we are going to prepare a plan such that who and all the programmers are going to be involved in this, and what domains we are going to give to which programmer. That particular plan we are going to come up with. That is called your programming plan. Once the programming plan is there, then we are going to actually start with program. That is, here is what we are going to write, the SAS program. Remember, whatever the program we have created, the output of this particular program is nothing but, that is SDTM output is nothing but, can I say SAS datasets? All of you? Because we are not going to ADaM yet, we are not going to your TLF yet. So at this particular level, the SDTM output is going to be SAS datasets. Am I correct? Every one of you accept that? The results are nothing but SDTM, the SAS datasets. Yes or no? Right. So, but remember, they cannot utilize, that is, the report cannot be in your SAS datasets. We need to convert this particular SAS datasets into one of the specific format. You can call it as XTT files. There are going to be bunch of softwares available to you, and by using those bunch of software, I'll tell you, I also help you all this information like how these XTT files are going to be. So we are going to convert your SAS datasets into XTT files. Then we are going to use different companies use different softwares, but one of the most commonly used software is going to be Pinnacle 21. By using the Pinnacle 21, we are going to use all these XPT files. We are going to say that these are going to be the Sdtm implementation guide 3.2 as per the 3.2 standards. We are going to create XML file. This is our final output, right? So this is called an XML file. It is always going to be define.xml, this particular file, and we also going to give validation notes. That is, we are going to validate this and we are going to produce this XML file. These 2 files we are going to submit to the, that is called submit study. These are the ones we are going to submit. So the first initial step is going to be get your protocol, get your CRH, and also have your SDTM implementation guide. Create your annotated CRF, which have information about domain and variable name. Then prepare your SDTM mapping specification, which has information about what variable, what table, what is the length, nothing but the metadata information. Once you have it, have your programming plan. What are all the domains involved, which programmer is working on which domain, and then start your programming. Once you complete your programming, you're going to have your output files. That is nothing but SAS datasets. Convert those SAS datasets into XPT files. Input these XPT files to the Pinnacle 21 software, which is going to create XML file. We generally call it as define, spelling mistake, define.xml file, and it also you need to create a validation notes. Both of them submit as your study results. Is that clear? What is called your SDTM is and what is going to be your workflow? Whatever you can see it on the screen, I'm going to put everything into Excel with a proper flow chart, and again I can submit to you or I can share it with you. Is that clear? First of all, on a very high level, on a day 1 of your SDTM, are you able to understand the workflow on a very high level? Yeah, Sanchi, we are not going that detailed here. True, but we are not going that detailed here. Any questions from anyone? Can I have a quick confirmation on this? Any questions from anyone? On a very high level, what we are doing, right? So what we are going to start then, let's say if this is going to be the workflow, what we are going to start. We are not going to start about how to create the CRF, what is the CRF and all. Instead, because our main thing is going to be identifying this, correct? I want to create SDTM mapping specification. For that, either you're going to get leverage on the previous programmer's data or something. If not, if you want to start from the beginning, you need to have a very good understanding about these domains, correct? What is each domain, what variables are part of it, you need to understand that, correct? So we are going to have different domains in SDTM. We have special purpose domain, we have interventions, we have events, and we have findings, right? Sometimes we can also say that trial design models also there, that is called TA, TST, TBTI. All these are these 1 2 3 4 5, the first 5 you can see, these are called trial design models. Okay, let's say if I say AE is part of your events. This one, this is events, right? And you can see there is something called MH. The MH is also part of the events. So you can see something called CM, right? CM is nothing but as part of your interventions, right? You can see something called other SDTM. There are so many other, I can give you the details as well. But on a nutshell, we are going to have the different domains. We start with the special purpose domains, which are very important, which are nothing but the DM, CO, SE, and SV. So those are the ones we are going to start with this. Yeah, core variable Raja, I can explain because it's already 9:15. We can discuss in detail because when you go with each domain, we are going to discuss about all this, not one, all this, right? In detail, what is each variable is, what label, and all the information. So no worries about it. I can explain that. Is that clear, guys? So tomorrow we are going to start with the SDTM guidelines first. There is going to be a guidelines, and then we are going to start discussing about each of this. That is nothing but what is code. Under the code we have, maybe I can show you one. So you can see REQ. REQ is nothing but required. Mean to say that these variables must be included in the domain. These variables cannot be null. That is, let's say study ID. I should not find any data set or any domain where my study ID is null. No, I cannot have it. So REQ is nothing but the required. And you can see something called EXP. EXP stands for expected, right? Meaning to say, these values supposed to be part of your data set, right? But they can be missing values as well. They can be missing values as well. That is, study ID cannot be a missing, right? There is no study ID which can be missing. There should be a value for it. But when it comes to the EXV, that is expected variable, if the core is going to be expected, this supposed to be part of my what to say, data set, but this value can be missing. Now there is something called PERM. PERM stands for permissible, right? It purely depends on the sponsor either to include or to drop, right? You can have it or you may not have it also, no issues at all, right? Even if you drop it, it's okay. The sponsor or the CRO are the one who is going to take this particular call, right? Raja, is that clear? On a very high level, but when you go with each domain, we can discuss that in detail. But you can see there are 3 values here. What are the 3 values? I'm trying to explain that. Correct? All right. So first we are going to discuss all the SDTM guidelines. So once the SDTM guidelines are there, then we can start with the special purpose domains. Once we start with the special purpose domain, our programming also going to start. Clear? So to start the programming, we need to wait for 3 more classes. So I can say that this week we are going to dedicate for SDTM guidelines. That is nothing but all the guidelines we need to understand. The guidelines include how to write your domain name, how to write your variable name, how I can say that what are going to be the various data types that are allowed, what are the formats that are going to be allowed, what is nothing but your core, what is nothing but the role, what are called identifier variables, timing variables, topic variables, and then we go with each of the domain. Clear? Right. So when is going to be the next class? So since it is going to be theoretical in nature, so today is going to be Wednesday, let's have class on tomorrow and day after tomorrow as well. That is nothing but you have classes tomorrow and day after tomorrow as well, right? So the next class is by tomorrow. Clear, everyone? So my target is going to be by day after tomorrow we can complete all the guideline, and then we can start kick off with our special purpose domains from Monday. Okay, so tomorrow and day after tomorrow we can complete all the guidelines. All of you, can I have a quick confirmation? Is that understandable? Harini, just get in touch with Balu sir, the person who you contacted, so he can help you with the recordings. Clear? Thanks everyone. See you tomorrow. Tomorrow there is a class. Please join. Thank you.